Okay, today we are going to talk about the citric acid cycle. So it is also called as TCA cycle. Okay, so or tricarboxylic acid cycle, Krebs cycle, the citric acid cycle, and the final common metabolic pathway. Okay, so final common metabolic pathway. So definition: the TCA cycle is the final common metabolic pathway for the oxidation of acetyl CoA, which is obtained from the carbohydrates, okay, lipids, proteins. So the TCA cycle is the final common metabolic pathway for the oxidation of acetyl CoA to carbon dioxide, water and energy. Talk about the site. Tissue site is going to be all the tissues of the body except RBC and lens, because these cells lack mitochondria, and the intracellular site is mitochondrial matrix. Then the starting compound, the TCA cycle, is going to be acetyl CoA, and the end product is going to be carbon dioxide, water and energy. Next, if we talk about the reaction pathway of TCA cycle. So we should write this reaction pathway in essays. So firstly, oxaloacetate is going to get converted to citrate in the presence of the citrate synthase enzyme. And then the citrate is going to get converted to cis-anconitate in the presence of anconitase enzyme. The cis-anconitate is going to get converted to isocitrate in the presence of same anconitase enzyme. This isocitrate is going to get converted to oxalosuccinate in the presence of isocitrate dehydrogenase. This oxalosuccinate is going to get converted to alpha ketoglutarate in the presence of isocitrate. This is DH, means dehydrogenase. This alpha ketoglutarate is going to get converted to succinyl CoA in the presence of alpha ketoglutarate dehydrogenase enzyme. And this succinyl CoA is going to get converted to succinate in the presence of succinate thiokinase. The succinate is going to get converted to fumarate in the presence of succinate dehydrogenase. The fumarate is going to get converted to malate by the fumarase enzyme, and the malate is going to get converted to oxaloacetate by malate dehydrogenase enzyme. So remember, if you forget all these intermediates, like if you forget all the byproducts, byproducts of the TCA cycle, remember to write the the enzymes and the intermediates of the TCA cycle. That will be enough. So firstly, this citric acid cycle includes the oxaloacetate, the citrate, the cis-aconitase, the isocitrate, the oxalosuccinate, the alpha ketoglutarate, the succinyl CoA, the succinate, the fumarate, the malate. So, and the enzymes. Remember to write the enzymes. So first enzyme is going to be the citrate synthase. Second is going to be the anconitase. Okay, similarly anconitase. Third is also anconitase. Isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, succinate thiokinase, succinate dehydrogenase, fumarase and malate. So initially, as you can see, the acetyl CoA, which is obtained from the carbohydrate proteins, lipids, proteins like amino acids, so are going to get the acetyl CoA, which is obtained from those, are going to get converted to this. Okay. After entering the TCA cycle, they will be converted to the CoA. Then we will talk about the inhibitors of the TCA cycle. The fluoroacetate, okay, firstly the fluoroacetate, the arsenate and the malonate are the inhibitors of TCA cycle. The fluoroacetate inhibits the enzyme anconitase. The isocitrate inhibits the enzyme alpha ketoglutarate dehydrogenase. The malonate inhibits the enzyme succinate dehydrogenase. So this inhibition is by first one, the fluoroacetate inhibits anconitase by non-competitive inhibition. The arsenate inhibits alpha ketoglutarate by non-competitive inhibition. The malonate inhibits succinate dehydrogenase by competitive inhibition. Next, if you talk about the energetics of the TCA cycle, next we will talk about the energetics of TCA cycle. During TCA cycle, 4 reducing equivalents are produced. So the 4 reducing equivalents are NADH, 3 NADH and 1 FADH. Remember, 1 NADH can produce 2.5 ATP and 1 FADH can produce 1.5 ATP. And 1 GTP is also produced. So we will talk about the steps, the coenzymes and the number of ATP produced. So by the end, the net gain of the energy, net gain of ATP in the TCA cycle is going to be 10 ATP will be produced. So first the isocitrate dehydrogenase will give rise to 2.5 ATP by the enzyme NADH, and the alpha ketoglutarate will also give rise to 2.5 ATP. The succinate thiokinase will give rise to 1 ATP. The succinate dehydrogenase will give rise to 1.5 ATP. The malonate dehydrogenase will give rise to 2.5 ATP. And you can study the coenzymes for these also. So remember to write the coenzymes for them. Then we will talk about the functions of the TCA cycle. The primary function of the TCA cycle is to provide energy. TCA cycle is amphibolic, that is both catabolic and anabolic in nature. So if we talk about the catabolic role, the TCA cycle is the final common metabolic pathway for the oxidation of acetyl-CoA. Okay, so acetyl-CoA, which is acetyl-CoA is obtained from the carbohydrates, proteins, lipids, proteins, amino acids, proteins like amino acids. So as you can see how this pathway is going to be there means, firstly, okay, the glucose is going to get converted to pyruvate. Okay, so pyruvate, the pyruvate is a glucogenic amino acid, and the pyruvate can produce acetyl-CoA. Acetyl-CoA is a ketogenic amino acid. Fatty acids can also give rise to acetyl-CoA, which enters this acetyl-CoA enters the TCA cycle to produce carbon dioxide, water and energy. So then we will talk about the anabolic role. So anabolic role, if you talk, means heme synthesizes. So firstly, it will be utilized the starting from the intermediates of TCA cycle. What is anabolic role means? The intermediates of TCA cycle, several compounds can be produced from the intermediates of TCA cycle. So for example, heme synthesis. The succinyl-CoA is used as a starting material for heme synthesis. The synthesis of non-essential amino acids like aspartate, glutamate. Okay, so oxaloacetate and alpha ketoglutarate, how these are formed means oxaloacetate and alpha ketoglutarate. Alpha ketoglutarate can give rise to aspartate and glutamate respectively. So alpha ketoglutarate will give rise to glutamate and oxaloacetate will give rise to aspartate. Then the fatty acid and the cholesterol synthesizes, and then the glucose synthesizes also. Intermediates of TCA cycle, so glucose synthesizes also. The intermediates of TCA cycle can be a source for the gluconeogenesis, can be a source for GLUCO, gluco, NEO, GENESIS, gluconeogenesis. What is gluconeogenesis means? The synthesis of glucose from non-carbohydrate source is called gluconeogenesis. Then we will talk about the regulators of TCA cycle, the citrate synthase, isocitrate dehydrogenase and the alpha ketoglutarate dehydrogenase. So these are the regulators of TCA cycle.